Southern blots are relatively ancient technology, but they still have a place in the world of molecular testing because they're the only method that gives us information about the local structure of the chromosomes near particular alleles that we're interested in. Information that's very difficult to get from methods like PCR or DNA sequencing. So they still have a place, and you can still order these tests. To understand how we get structural information as well as copy number information from a southern blot, we need to understand a few different uh, attributes of the method. So this is a slide taken from Dr. Geordie's book, where we've got two copies of each of our chromosomes. So let's call this chromosome 16. Whatever it is, we've got a maternal and a paternal copy labeled 1 and 2 here, and we should have very similar copies of gene A and very similar copies of gene B on the two homologs. But it turns out that they aren't identical to one another, and their context being slightly different means that we can use Southern blot to independently assess the copy number of allele A from chromosome 1 and allele A from chromosome 2, as well as B from chromosome 1 and B from chromosome 2. And what we'll get out at the end is this information over here, where we can then look and see whether either one of them has been affected without interference from information from the other allele. So before we talk about how a southern blot works, we need to understand three things about three tools that we have to examine our DNA molecules. The first one actually comes from bacterial cells, where they have sort of a primitive self-recognition system called restriction endonucleases. So these are enzymes. Restriction endonucleases are these guys that can recognize sequences in DNA. So they're going to cut up the DNA, but they're only going to do it when they recognize a specific sequence of the DNA. Oftentimes, the one we use in the lab have 6 base pair recognition sequences like this, and you'll notice that they're palindromes. So GGATCC 5' to 3' that way is the same thing as pairs to its hybrid is GGATCC going the other way 5' to 3'. So palindromic sequences are probably helping these enzymes recognize sequence. I just wanted to point it out. It's not an important feature of the system. What is important is that it only recognizes that 6 base pairs, no other sequence, not even one mismatch tolerated. And whenever it finds it, it will cut that and break the DNA molecule into 2 pieces, leaving a double-strand break that will leave the DNA in 2 pieces. The second concept we need to understand is how we can separate macromolecules by electrophoresis. So DNA molecules have a very strong negative charge on them because of the phosphodiester backbone. So, and because that's a repeating feature of the structure of the DNA, then all DNA molecules have not only a very large negative charge, but they have a relatively constant charge to mass ratio. So DNA molecules, every time I put in one more base, have a phosphodiester backbone, and therefore we're going to have a fairly significant negative charge, and it'll always be about the same. Every DNA molecule that's 1,000 base pairs long will have about the same ratio of charge to mass. That's important because that's what separates things in electrophoresis. So what we're going to do is we're going to take the DNA molecules and we're going to make a semi-solid matrix. It's not the same stuff as Jell-O, but it behaves identically to it. So you take this powder, you boil it up in water or buffer, and then you pour it into a mold. The mold will be a little bathtub-shaped thing here. This is about 4 inches long, and it's got a little divot in it right here. So we leave a little divot in the mold so that when we lay this out on the counter, there will be a place to put a liquid sample. So this is lying horizontal on a countertop. We put the DNA sample in here in that well, and then we apply an electric current to this to the gel. It's usually made out of agarose, or it can be polyacrylamide. It doesn't really matter what it is. The point is the DNA molecules have to move through this matrix, and they're charge is going to drive them forward because there's a current flowing, and they're charged to mass. The mass is going to hold them back, so the charge to mass ratio is what's going to separate things. Since they all have about the same charge to mass ratio and they have different masses, they're going to migrate at different rates. So we're going to have small things that are going to get through the matrix very quickly and easily. So they'll be down here after we've electrophoresed for a while, a couple of hours typically, maybe an hour. And we're going to separate these guys by size. So small things will run fast, and the positive electrodes down at the bottom, so it's going to attract the negative things. So all the DNA molecules are going to migrate into the gel. So over here on the left, we've got a sample that has just known size molecules of DNA, and they are separating out on a ladder because they are just different sizes of molecule that we put on there so we can tell how big the other molecules are. And over here on the right, we've just got a genomic sample that we've chopped up into little pieces. And since there are so many of them, we've taken a whole genome from an organism and cut it up with a restriction endonuclease. We have lots and lots of bands of different sizes, so it just looks like a smear. But we have within that smear separated all the molecules by size. The third concept we need is to understand annealing. So we've already talked about this. If I take two molecules of DNA that are antiparallel and complementary to one another, like over here, and I take them apart by denaturing them, heating them up, or changing the pH, or whatever I do to get those two strands apart, then when I reverse those conditions, say I cool it back down, or I take away or change the pH back to neutral, then they're going to try to find each other in the antiparallel complements. So this process of hybridization is relatively slow, but these molecules can sort this out and say, okay, you're the wrong thing, you're the wrong thing, that's not going to work out, that's not going to work out. This looks promising. I have found an anti-parallel complementarity group here with C and C prime, and they're quickly going to figure out that the rest of these guys also belong together, and they're going to zipper up and give me back my molecule. So this is the basis of hybridization, is that nucleic acids can find each other because of their stability of anti-parallel complements relative to the instability of things that don't have anti-parallel complementarity. So I can take advantage of this by taking one molecule in a gel and asking a probe molecule to go find the place in a gel where its antiparallel complement is going to be found. So this hybridization property is the third thing we need to know before understanding the southern blot, and now we can look at how they work. So putting that all into motion, let's take a look at the starting material. We started off with two chromosomes. Instead of labeling them 1 and 2 like they were in the first slide, I'm going to call them mom and dad because that's who we inherited it from. We have two homologous chromosomes, one we inherited from our mother, one we inherited from our father. And now we're interested in figuring out how many different alleles we have in a particular area. So we're going to assay both the copy number and the structure of the chromosomes in a particular region in the area of this probe. So this method is only going to tell us about a specific area that we are interested in. So we have to already know the area we're interested in. But it could be a fairly large region, 5,000 or 10,000 base pairs long. So once we're interested in that region, the only way we're going to get more information from a southern blot than to find out what the copy number is we have to have what are called restriction fragment length polymorphisms, or RFLPs. So going back to the concept of restriction enzyme digestions, remember we had restriction endonucleases that recognize 6 base pair sequences. There's one there, there's one there. Here's that restriction endonuclease site is found in both the mom and dad versions of the chromosome, as is this one down here. So we've taken some restriction endonuclease. Its 6 base pair recognition site shows up more or less at random in our chromosomes, but sometimes there's a base pair change, a single nucleotide polymorphism, the SNPs that we've talked about. But in this context, because they change whether or not a restriction endonuclease can recognize something, we call them restriction fragment length polymorphisms, or RFLPs, because, for example, this guy right here, that restriction endonuclease site only shows up in mom. It's not going to show up in dad. Uh, so now if I start cutting this DNA up with those restriction endonucleases, I'm going to get different sizes of fragments. So mom over here, if I digest up her chromosomal DNA, I'm going to get, in the area of the probe, I'm going to get one short piece here and one long piece here. If I cut up dad's DNA in the same region, I'm going to get an even shorter piece over here and an even longer piece over here in the area of the probe, because I'm going to cut the DNA with the restriction endonuclease, and it's going to give me a double strand break there, a double strand break wherever there's an endonuclease site. So I'm going to cut the DNA up into different sized pieces in this area. And that's sort of the crucial concept at the center of the southern blot, is that the restriction endonuclease cuts the DNA up into different sized fragments if there are restriction fragment length polymorphisms in the area. And I come in and ask, how big is that piece of DNA with my hybridization probe? Then it's going to tell me that I've got different sized fragments. If the restriction endonucleases are all the same, say if I were to probe in this region, I would always get the same answer. That piece of DNA is that big. So I wouldn't be able to tell the alleles apart over there. But here I have a bunch of restriction fragment length polymorphisms in this area. So I'm going to get a very rich idea of which allele I'm inheriting from both mom and dad when I look at their child's southern blot. So to do the southern blot, we first do the restriction endonuclease site, the restriction endonuclease digestion. So that's going to be the first step here. I take the DNA from the mom and the dad and the kid, and I digest it up with the restriction endonuclease. Second step is I run it out on an agarose gel using that electrophoresis method I just showed you. And that means I'm going to get separation of the DNA molecules by size. So the small things are going to run faster, the large things are going to run slower. So as we said, mom's going to give us two intermediate sized pieces in the region of the probe, that one and that one. Dad's going to give us a bigger one and a smaller one. And whatever molecules the kid inherits, we will now be able to tell which chromosome was inherited. For whatever reason we're interested in that, we will now be able to tell which allele it was inherited by the child. We can't see those molecules. I've labeled them here in blue and red, but of course we can't really see them. All I get when I run out the whole genome's worth of DNA is a smear, because there are several hundred thousand or a million fragments, depending on the restriction endonuclease. Then there are going to be many, many fragments of DNA, and they're not going to separate. I won't be able to see individual bands. So the concept of the southern blot was that I could take the information that I obtained from the two-dimensional separation of the molecules by size, retain that information, and then ask, using a hybridization method, where those molecules went by probing them with a specific hybridization probe. So to do that, after I have separated the molecules in my agarose gel, I need to transfer them onto a membrane so that I can probe for the presence of particular DNA molecules. And that's the blot part. I'm going to put a membrane on top of the agarose gel, and then I'm going to put blotting paper behind it. The liquid in the gel is going to move out from the gel through the membrane, and as it goes, it's going to carry the DNA molecules, but they're going to stick on the membrane. So they're not going to get through. I'm going to blot the liquid out, but I'm going to trap the DNA molecules on the membrane. So now the membrane is going to have all the two-dimensional information that tells me the size of the DNA molecules, and it's still going to be retained on the membrane. Okay, so the DNA is now stuck to that membrane in the same pattern that it was in the agarose gel, and now I have to do my hybridization. So going back to the hybridization step, now I denature all of my DNA molecules on the gel. I take my probe molecule, which I have purified some DNA from somewhere, and I denature it, and I label it somehow. Radioactivity, fluorescent dye, whatever I do, the same sort of tools that we've always used for in all of our probing methods are available here. And I take a bunch of those little purified DNA fragments and I mix them with the blot, and I let them run around and do their hybridization. One of the molecules, the probe, is free in solution. The other one is stuck to the membrane. But it's the same process that I showed before, where we get nucleic acid molecules to stick to one another by figuring out where their complementary antiparallel complement is. Okay, so that all happens on the surface of this membrane, and then I just wash away everything that doesn't stick. So all the things that weren't antiparallel complements, I wash them away, and now I only get hybridization of the probe molecules to specific places, and I can detect them. Autoradiography to find P32, a fluorescent scanner to find fluorescent dyes. Whatever it is, I'm now detecting the probe molecule only. And then I get my southern blot, which tells me how big the DNA molecules were in the mom and the dad and the kid, and I can then infer which allele was inherited by the child, or whatever it is I'm interested in. So that's how the southern blot works. Now let's look a little bit at how we choose a probe. So going back to our original figure, what we have now is we've got a normal cell and a tumor cell, and we have two copies again, 1 and 2 of our two chromosomes, of this same chromosome, and I've got two regions on it, one on one arm called A and one on the other arm called B. So they both have restriction fragment length polymorphism, so I can independently assess whether I've inherited or I have the presence of A1, A2, B1, and B2 using a southern blot. So let's imagine what the genome must look like in order to give me the result that I'm seeing on the left. So if I inherited A1 from mom over here, she must have had restriction endonuclease site patterns like so. The probe I'm going to use is this region here, so I'm going to take DNA from that region and use it to hybridize to my southern blot. So that's the only region of the genome I'm going to see. When I cut with the restriction endonuclease, A1 and A2 are going to give me different size fragments because I have a piece this big from A1, but a piece this big. There's another restriction endonuclease site that isn't found in A1. Therefore, I'm going to get a large fragment and a small fragment from A1 and A2. The same thing is going on with B1. So when I use this probe to detect a region of B1 that I'm interested in, of B that I'm interested in, I'm going to get this size fragment from B1, and I'm going to get a shorter fragment from B2 over here, because there's an extra restriction on the nuclease site. It doesn't matter that there's a little bit of stuff over here that doesn't show up in the probe. That molecule is still going to hybridize to my probe, so I'm going to see it. It's not going to hybridize to every part of it, but the fact is it's only going to show up that molecule, and because it has complementarity, it is going to show up. So this is what a map of the region must look like. I've got two RFLPs, a short and a long fragment from A1 and A2, and now when I do my southern blot, I can independently detect them. So in an original normal cell, I'm going to have both A1 and A2, so I'm going to get this result over here where my normal cells have two bands. So the normal A and the normal B both show up as two bands because I've got these two fragments there and there, there and there, and they're both showing, all four of those guys are showing up. When I go to the tumor sample, I'm still getting both copies of A because A is normal in the tumor sample over here. But B has lost one of the chromosome arms, so we have had a deletion of an entire arm of a chromosome over here. So now I'm only going to get B2, but I'm not going to get any signal from B1, so it's missing over here. So this is diagnostic for a chromosomal deletion and tells me that that when I went from normal to tumor, one of the genetic hits that I took was that I suffered a deletion of that part of that chromosome. So that's what we can learn from the southern blot. We can tell what the copy number is, but we can also tell something about the structure of the genome in the local area around a probe. So that's why it's still useful. If I just wanted to know copy number, again, I have better ways to do that. So this wouldn't be used just to find copy number, but it is really one of the only methods that allow us to determine the structure of the genome by using restriction endonuclease polymorphisms, so that I can tell which allele I'm looking at a particular time. So that's why southern blots are still around, and this is why we would use them in medical diagnosis.